Manual tissue dissociation is often labor-intensive, time-consuming, and inconsistent. Variability in enzymatic and mechanical processing can affect cell viability, yield, and reproducibility, making it difficult to reliably generate single-cell suspensions. To simplify tissue dissociation and improve consistency, STEMCELL Technologies developed STEMprep. This automated system standardizes processing, ensuring high yields of viable single cells from a wide range of solid tissues while maintaining reproducibility. By automating homogenization and dissociation, STEMprep reduces manual effort, preserves epitope integrity, and delivers high-quality single-cell suspensions, helping researchers focus on what matters most: their downstream experiments. With an intuitive touchscreen interface, STEMprep simplifies protocol selection and setup, streamlining workflows. Built-in temperature control from 4 to 37 degrees Celsius ensures optimal sample processing for a wide range of sample types and applications. STEMprep grows with your research. Begin with a primary unit to process up to 4 samples at a time, and expand with secondary units to scale up, enhancing throughput while maintaining efficiency. Use validated stem cell protocols or create your own, enjoying the flexibility to adapt STEMprep to different tissue types and experimental needs. With automated precision, temperature control, and scalable flexibility, STEMprep ensures efficient tissue dissociation with high yields and reproducibility. Discover how STEMprep can enhance your tissue processing workflow. Visit stemcell.com/STEMprep.